I don't wanna grow up, I'm a curious kid. The world has 100 questions I can play with. So I'll open my arms and eyes and wonder every day till the day I die. No one really knows why gravity exists.
I want to introduce UC Berkeley psychologist Antonia Langenhoff on what childhood teaches us about human uniqueness. Antonia earned her bachelor's and master's degrees at the University of Göttingen in Germany, and then came to Berkeley to do PhD research in the developmental psychology lab of Professor Jan Engelman, himself a Wonderfest speaker from just a year ago. Please welcome psychologist and Wonderfest Science Envoy, Antonia Langenhoff. There you go. Please share your slides, Antonia. There we go.
Thank you for that nice introduction. All right, can you see my slides now?
Yes, indeed.
Wonderful. Okay, yeah. Thanks for that great introduction. I'm indeed a psychology PhD candidate at Berkeley. And as Taco already said, I want to introduce you to the world of developmental psychology today and show you what studying children can teach us about human uniqueness. So, did you know that we share almost 99% of our DNA with chimpanzees? That's because our species and chimpanzees diverged from a common ancestor about 8 to 6 million years ago. It might sound like a really long time, but in evolutionary terms, it's really nothing. And as a result of that, there are a lot of things that we, humans and chimpanzees, have in common. So, unlike most other animal species, humans and chimpanzees both love to play. They display complex emotions, and they invest a lot of time into raising their children. Nevertheless, humans are the only species that flies to space, invents vaccines, and uses complex technology. Why is that? What makes human minds and human lives so different from those of other animals, including chimpanzees? Now, there are many ways to answer this question. And one intuitive response that people often give is that clearly humans are smarter, more intelligent than chimps, right? But that's not necessarily true. For example, many chimps have an incredibly strong working memory and can easily outperform many humans on memory tasks. Chimpanzees are also very competent tool users. They can build elaborate tools to extract food and water from almost anywhere. So from that perspective, allow me to say that individually, at least, most humans may not be that much smarter than the average chimp. Now, here is my answer to this question. I think that also humans aren't necessarily individually smarter than chimpanzees. They are smarter in groups. I, like many others in my field, think that humans have unique social and cooperative skills that allow us to put our heads together to create things that are bigger and better than what we could have come up with on our own. And that's what makes us special. Now, crucially, many of these remarkable social and cooperative skills emerge very early on in life. And today I want to give you a taste of the research in developmental psychology that shows that. Okay, first, helping. So let me show you a video from an influential study by Felix Warneken, a professor at the University of Michigan, showing helping in very young children. Oops, sometimes this video is a little funny. Give me one sec.
Oh. Oh. So?
All right, so let me note a few things here. First, a super quick comment on methodology. So what developmental psychologists do is not just looking at cute videos of kids and deciding that what the child is doing there is, for example, helping. We specify a priori hypotheses and about what we expect children to do. And we define exactly what counts as, for example, helping and what doesn't count as helping in these kinds of situations. And then we test many, many, many kids in the same situation. And then we analyze the data we obtain in this way using statistical models and draw conclusions based on the results. But since we only have 20 minutes today, I decided mostly to focus on the cute videos, but just so you know. Now, a second comment here is that I should mention that chimpanzees help too. So for example, they will pick up an item that their caregiver dropped or something like that. But children help way more often and in a much wider variety of situations than chimps. And what's more is that human children seem to inherently enjoy helping. Now, the third point I wanted to make here is that the children in this study and in other similar studies were often only 18 months old, so they were still pre-verbal. And nevertheless, they read the situations like the one you just saw in similar situations with ease, and did exactly what was required to help the experimenter in the particular situation. So spontaneous helping really emerges remarkably early on in development. Okay, here's another example for an early emerging human social skill, sharing. So let me show you a video that's not directly from a research study, but I think still tells us a lot about early sharing behaviors.
I'm gonna go and I'll be back in 5 minutes. Okay? Here I go. Count to 3.
1, 2, 3. There. I don't know. This one or this one? This one. And let's do magic. The bigger one. Yum, yum.
Okay, so what the kids in this video do is very much in line with research investigating children's sharing behaviors. This research shows that up until age 3 or 4 or so, children often behave quite selfishly. But after that, children develop a strong preference for equal resource distributions. They share their own resources, especially in situations like the one you just saw, where one child sort of randomly gets more than the other. And they also expect others to do the same, and they protest if others don't share. Now, these are just the very basics. If you're interested, there's a whole range of interesting findings out there related to early sharing behaviors, but I won't dive into that any deeper today. Instead, for the rest of my talk, I want to focus on a social skill that's a bit more complex than helping or sharing, but if you ask me, even more important in understanding what makes humans special, which is why I decided to study it for my PhD. And this skill is called collaborative reasoning. Now, what does that mean? So I think it's safe to say that none of those amazing human inventions like spaceships, vaccines, or computers would have been possible if a single person would have tried to come up with them on their own. These inventions were all a product of multiple people putting their heads together, exchanging ideas, and arguing about the pros and cons of different alternatives. And that's exactly what collaborative reasoning is. But I think collaborative reasoning doesn't only happen when people design spaceships or invent vaccines. I think people do it all the time and from early on in life. So together with the other researchers in my lab, I aim to uncover when and how exactly collaborative reasoning develops in young children. And we do that by working with children not only in the US, but also in Kenya and in China, because we think it's important to understand both similarities and differences in the development of this ability across cultures. Okay, let me tell you about one of the studies I'm currently working on. It's still in its very early stages, but it's the study I'm currently most excited about, so I wanted to share it with you. Okay, these are all the people involved in this study. So as you can see, we don't just study collaborative reasoning in children, we also reason collaboratively ourselves. And here's a little bit of background on the study. So one way to solve disagreements is to solve them competitively, using force or dominance. The latest tragic reminder of that on the political level being Putin's war on the Ukraine. Now, an alternative, better way to solve disagreements is to solve them cooperatively through collaborative reasoning. Or, as the German philosopher Jürgen Habermas put it, through the unforced force of the better argument. Now, in our study, we're interested to see whether children make use of this unforced force of the better argument in situations of joint decision-making. Specifically, what we want to know is if children resolve disagreements with others by giving reasons. And to do that, we test children who are aged 5 to 9 and who live in Berkeley, the US, in Laikipia County in Kenya, and in Beijing, China. And here's our study setup. So 2 children have to decide together where a reward, 2 stickers for one for each child, is hidden. For example, whether they're hidden in the yellow box or in the blue box that you see here. Now, before children get to make their decision together, they individually receive information about the reward's location. So, for example, one child is told by the first experimenter that the stickers are hidden in the blue box. And at the same time, the second child observes how a second experimenter places the reward in the yellow box. Now, after children receive this information, the experimenters leave the room, and children get 60 seconds to decide together which box they want to pick. They can only pick one. And what we expect to see here is collaborative reasoning in action. So we expect that children will find out pretty quickly that they disagree about which box they should pick. And we expect that that will lead children from all 3 cultural backgrounds to give reasons for their respective beliefs about where the stickers are. So the child that was told that the stickers are in the blue box should say something like, I think we should pick the blue box because she told me they're in there. And the other child that saw how the stickers were placed in the yellow box should say something like, no, we should pick the yellow box because I saw her put them in there. So what we're measuring in this study is children's spontaneous reason giving. And to quantify that, we compare how many reasons children give in this disagreement situation to how many reasons children give in an agreement context, where both children think the stickers are in the same box. And we expect, of course, that children will give more reasons in the disagreement situation than in the agreement situation. Now, we've only tested a few kids so far. Like I said, this study is still in its early stages, but let me show you some examples. So here's an example from Berkeley. Sorry about the watermarks here. I just wanted to add subtitles. This is an example from the disagreement.
Yellow. She said that it's in the green, so mine said it was in the yellow. She put them in the yellow. I saw her put it in. Okay, yellow. Okay, yellow was in yellow.
Okay, well. All right, so what I find hilarious here is how quickly and efficiently those 2 make their decision, right? So right upon noticing that they disagree, each child immediately shares their reason for the respective belief. And once the reasons are both out on the table, so to speak, they immediately agree on the yellow box. So what this implies is that at least for Berkeley children, seeing where the stickers were placed is a better reason than being told where the stickers were placed. That's collaborative reasoning at its best. Okay, here is the second example from Beijing, China. This is again the disagreement context.
可是阿姨给我说的是紫的，我是吧？你真的看见了啊？我觉得他们又会学这个。我真看见黄的，做错了。刚才那个哥哥直接把这个放在黄色的盒子里，然后就把盖让我回来，然后我回来了。
Okay, I loved watching this video even without understanding what these two are actually saying there, because you can clearly see how excited they are. And you know, from reading the subtitles, I hope you could tell that, like the children in Berkeley, both of these girls provided a reason for their belief, right? The one girl said, the teacher told me it was in there. And then the other girl said, no, it's yellow. I saw it with my own eyes. So at this point, I unfortunately don't have any videos from Kenya quite yet for data protection reasons. I can't show them to you, but I can tell you that the Kenyan children our collaborators have tested so far also give reasons to support their beliefs in the disagreement context. In contrast, like the children in Berkeley and in the US and in China, those kids in Kenya hardly ever provide reasons in the agreement situation. So taken together, our very preliminary evidence suggests that by age 6 or so, children in all three cultures, in the US, in China, and in Kenya, are avid and adept collaborative reasoners, despite the different circumstances in which they grow up. And I'm very excited to see how this project continues in the future. Okay, so I'm getting close to 20 minutes here, so I'll end my little tour through research on human socio-cognitive development here. Now, there's a whole lot of other research out there, way more than I could fit into a 20-minute talk. But I hope that by showing you how children help others, share with others, and reason collaboratively, I've convinced you that humans are social and cooperative beings from early on in life. Although, when looking at all the antisocial and uncooperative things adult humans do all the time, it seems like we sometimes need to remind ourselves of those early cooperative roots. Great. With that, I thank you very much for listening. Yeah, and I'm happy to answer any questions. Thank you.
Thank you, Antonia. I see we do have a question already. I'm not sure whether our anonymous Zoom user wants to ask the question himself. Not seeing a raised hand with the reactions button at the bottom of the Zoom window. I'll suppose that he or she is going to let me read the question aloud. Antonia, how long ago do you think humans developed their unique social and cooperative skills? Prior to Homo sapiens, Erectus, or Habilis, or later?
Yeah, that's a great question. So I'm not an anthropologist, so this is a little bit out of my expertise, so to speak. But there is a lot of people out there who think that it probably was Homo sapiens who developed these unique skills. And there's a theory out there, and I mean, like any other evolutionary theory, it's a little bit hard, to prove. But there's a theory out there that says that these skills were probably a product of humans changing their sort of like day-to-day life because of some sort of external constraints that they had. So there wasn't as much food available as there used to be. So the story goes. So humans had to sort of like find a new niche. They had to sort of like find food, and they went for the food that they could hunt and get in groups. So during that time, they sort of developed these unique socio-cognitive skills that would allow them to sort of go for the food that was only possible to get by working together, by sort of through collaborative reasoning, if you want, by like developing a trap, or by like hunting down a deer and then getting at it from different angles and so on and so forth. So that's the idea. That is sort of one explanation for why humans may have developed these kinds of skills in the past. And that, I think, was Homo sapiens.
Thanks, Antonia. I don't see a hand up or another question, so maybe you'll let me ask a question here. Early on, you talked about children helping more often than chimps help, and that evidently they enjoy that helping more. Can you describe that evidence? Are there some numbers here? Is it children help 90% of the time, and chimps hardly ever, or is there some overlap where chimps will come through some of the time?
Yeah, I don't have the exact percentages, but this is definitely the case. So there's one sort of influential paper out there that really compared human and chimp helping behaviors in sort of similar tasks. And, all of these tasks, like the one you saw in the video earlier, were nonverbal. So they really tried, and, you can talk about that too and have your doubts, but what they tried to do was to come up with situations that were sort of like equally easily interpretable for both chimps and human children. And in these tasks, human children helped a lot more often, significantly more often, than chimps. So pretty much human children help pretty much all of the time. And in chimps, they did see helping behavior in some of the tasks, but really not in most of them. And that was a significant difference that they found there.
Okay.
And this is from one paper that actually specifically contrasted the helping behaviors of those two.
All right. Thanks, Antonia. In that adorable video of children sharing, I guess, a peanut butter and jelly sandwich.
I assume so, yeah.
In every case, with one child receiving the sandwich and the other having the empty food dish, we saw sharing happen, and it was quite touching. Do you happen to know whether there were any children that didn't share?
I don't know about this case. Like I said, it wasn't from an actual research study, but just sort of a do-it-yourself at-home experiment.
Yeah.
But from research and sharing, for sure, like there's always children who don't share, but the vast majority of them does share. And then I didn't get into this, but there are of course also factors that affect whether children as a group share or do not share. So the situation you saw was one where it was very sort of like random or arbitrary, right? Which of the two kids got more or got the sandwich, and which of the two kids didn't get the sandwich. But we also find that, for example, if one child has worked really hard for getting a certain resource and another child hasn't, then at the same age, 4 years or so, children develop this intuitive assumption that if somebody worked to earn a certain resource, they should also keep it, and they don't necessarily have to share with the person who didn't work for their resource. So there's a whole lot of, whole range of factors that affect sharing behaviors as well. But in situations like the one you saw, most children, like the vast majority of them, will share.
Oh, neat. All right. Thanks, Antonia. I see Stuart. Welcome back, Stuart. You have a question. Often you like to present it yourself. I'll be happy to read it, but would you like, I see no hand. All right. Stuart asks, are you aware of any conflicting social or environmental situations and that you studied perhaps, which inhibit such collaboration among children, such as maybe home issues, societal upheaval, etc.?
Yeah, I think you're getting at, that's a really great point. So I'm really interested in that myself. Maybe not so much home issues, so sort of like the background of the children. There's also people who study that, although I'm not super sure if children's socioeconomic backgrounds, for example, have been linked explicitly to their collaborative behaviors. So I'm not aware of that research, but I would assume it's out there. But I think more broadly, factors that affect whether children collaborate, help, share, and so on and so forth is something that I'm really interested in. And I think, whether we do these kinds of things also greatly varies based on the context we're in. So, think about collaborative reasoning, right? What I showed you was an example where kids are being really collaborative and they're really interested in finding out where the stickers are hidden together with the other child. But there might also be situations where, for example, one child doesn't want to collaborate with the other child and is interested in maybe deceiving the other child, or where one child has strong other motivations to not collaborate, and so on and so forth. So I think the specific social context and the social motivations we have in a certain situation really affect these kinds of collaborative behaviors. And that's something that people are only just starting to do research on. And I think that's something that is super, super important, because, when we create these situations, we come up with these sort of like clean situations that we have in the lab, but then in everyday life, these kinds of things look very different often, and we need to understand why. And I think, in answer to this question, why do people often not collaborate in real life, is, well, there are certain other motivations and certain other factors that come into play. And I think we're just starting to systematically understand what these kinds of factors may be.
I gotta suppose that the hunger challenge, the hunger issue might appear in the sandwich sharing question. If a child is truly famished, maybe then sharing won't be quite as frequent or as generous when it does happen. Thanks for
and
your
children
question.
understand that too. So children are going to distribute more resources to other children who display a need, for example, who are hungry. That's actually something that has been shown too.
Great. All right, Antonia, thank you. Let's see, I'm monitoring the chat here. No more questions. I see well, I just want to read a comment here. This is a very exciting presentation. Thank you. Where may I find resources like pictures or books and video clips to show picture books, video clips to show kids about cooperation? I am working with K through fourth graders, kindergarten through fourth graders. Do you have any resource recommendations, Antonia?
Yeah, I saw that comment and I started thinking about it. Since my target audience is not fourth graders usually, but adults, I honestly don't know. That's a great question, and I wish there was something like that out there. I mean, those videos that I just showed you, at least one of them, you can find them on YouTube. But other than that, I can't think of anything. It sounds like something that we really need. One thing you could do is you could also run sort of experiments, kind of like the sharing one that I showed you. You could do that with your fourth graders, right? And you could see what they do. I feel like that would be pretty interesting.
Very good. Stuart, I think you have one more question. Let's make that the last one for Antonia. Please fire away, Stuart.
Yes, hi. Thank you for the previous answer and your presentation. Very much enjoyed it. Besides the environmental effects that you kind of answered previously, do you have any indications of maybe a genetic inclination against sharing? I think I came across something that said, a significant portion, like 1 or 2% of or more of our population, are actually genetically psychopath. And so I don't know whether or not that would show up at this young age. But any indication on that, or?
That's really not something that I look into in my research, but yeah, I mean, good point. I'm sure, and besides being a psychopath or somewhere on that spectrum, there's probably other inter-individual differences, right? That's what you're getting at, where that sort of like makes people not show these collaborative or social behaviors that I was talking about here, and I agree. There's probably other examples as well. And I'm sure that if you would really sort of dig into individual differences, you could find, differences in, for example, sharing even at a very young age. My sort of approach is to, look at what a whole range of kids do in these kinds of situations and to focus more on similarities than on inter-individual differences. But I'm sure if you looked into it systematically, you could find that as well. Yeah, that's a good point.
And how they might actually be indicative of the kind of person this child will grow up into, right?
Absolutely. Yeah, totally.
Great, Stuart. Thank you. Antonia, thank you. As Paul prepares to share his slides, what if our audience gives you an American Sign Language round of applause? Yes, all right.
Yeah.
Thank you, Antonia. You'll get applause, as will Paul, at the very end of this session. But Paul, please go ahead and share, and I will introduce you. Let's see. Very good. All right, now Stanford marine biologist Paul Bump will present Underwater Metamorphosis, a larva's tale. Paul earned his undergraduate degree at the University of Hawaii, the Manoa campus, and then spent 2 years working on stem cell research at USC. Paul now does his PhD work at Hopkins Marine Station on Monterey Bay. That's a Stanford outfit. He has long loved science outreach, as evinced by a TEDx talk he gave 5 years ago. Please welcome Stanford marine biologist and Wonderfest Science Envoy, Paul Bump.
Thanks, Tucker. Thanks for the introduction tonight and also for running this program for the Wonderfest Envoys. It's been a real pleasure to continue to work on my science communication skills, and I'm excited to try them out again tonight. You guys can let me know how effective or not that they are. But before I jump into my research, I just wanted to give you sort of a quick context. So Tucker mentioned I'm a student at the Hopkins Marine Station. So this is the 6th year of my PhD. So I'm almost done. I'm defending in May, so getting near the end. It's been a really enjoyable experience getting to explore the natural world, both in the beautiful sort of natural environment of Monterey Bay, but also here in the research lab where I spend far too much of my time. But also I think it's important to tell you, like, what does a PhD student actually do? And so we do a lot of research, but I would also say, you know, a real highlight for me in my PhD is by studying a diversity of organisms. I study a lot of different animals. I'll tell you about one of them tonight, but we typically actually work with a lot of different animals, and I think that's so great for expanding your mind, and so we can talk more about that later. But also a real highlight for me as a PhD student has been getting to build community with other scientists and just build that scientific exchange, and hopefully give you guys a piece of that tonight as we sort of share the science that we do. And also just the teaching and the mentoring that we get to do. So that's a little bit very quickly about who I am, but what I'm here to do tonight is to tell you a story about metamorphosis. We can get into exactly what a larva's tail is. It's like a pun, but it's a complicated pun. We don't need to get into that. But essentially, metamorphosis as a topic is uniquely fascinating, I think. I'm excited to tell you about this animal. It looks like on this slide that there may be 2 animals on the slide, but in fact, it's the same animal, just at different stages of its life. And so we'll talk about how exactly that occurs in this animal's lifespan. So the ocean is full of amazing animals. I think this is a thing that fascinates biologists, but also hopefully everyone in this audience. There's so many beautiful, intricate, and crazy wild-looking animals in the ocean. The thing is, they also have incredibly fascinating, complicated, alien-looking larvae. So all of these animals, I'm going to show you their corresponding larval forms, which to me look nothing like their adults. You can take, for example, this coral. When you think of a coral, you think of branching reefs. Well, it starts out as this tiny little bullet-shaped thing, very, very small. It's a planula larva. These are sea urchin larvae. They look like tiny spaceships. Like, did they come from outer space before they turned into sea urchins? Like, I don't know. So all of these animals have incredibly unique larval forms that are often quite distinct or different than their adult forms. And that's sort of what motivates me as a biologist. Is you see this diversity of shape and form and function. How does that actually happen? It's pretty wild. In terms of building those adult forms, there are sort of 2 broad main ways that animals do this. So one way, which we're probably most familiar with, is this idea where you go from an embryo to the adult. So this is a time-lapse video of zebrafish development. Zebrafish is a great model for understanding vertebrate development. So in this video, you're going to see this ball of cells eventually turn into something that looks a little fishy. That's similar to the type of development that we undergo as humans, that mice undergo, that a lot of animals undergo, and specifically animals that we often study in the research lab. There's good reasons for doing this. Animals have direct development. They often form the adult relatively quickly. Some species reproduce relatively fast in the lab. There's a lot of good practical reasons for studying animals with direct development. However, if we study animals with just direct development, we miss a whole other swath of animal diversity, specifically those with a very creatively named indirect development. So here's a sort of drastic example of this. So this is kind of a crazy video in which what is the adult form of this worm is actually eating its larva. So it first was born in this swimming little larval form, but now it's actually eating the larva that it came from. Now, this is like a sort of extreme example of indirect development, but it happens. There's all sorts of wild forms of indirect development. They sort of all get lumped together. The one you're probably most familiar with is butterflies, for example. You know, if you see a little caterpillar turn into a cocoon or a chrysalis and then emerges as the adult butterfly, that's a form of indirect development, a way of forming the adult, not directly from an embryo. Sea urchins, I mentioned their crazy little spaceship thing. In this particular example, the adult is formed from this little pocket of cells that emerge on the left side of the larva. That will end up forming this big spiny ball. Again, very different larval and adult forms. The third example is maybe a little more transformational, I'll call it. And so in this case, you have this sort of funny little shaped, it's called a trochophore larva. It forms its adult sort of by extending portions of the larval body and modifying some things. So it's a little less extreme than, say, I don't know, eating the house that you came from. So there's clearly a wide range of strategies. And the type of metamorphosis I'm particularly interested in are these transformational metamorphoses. Why I'm interested in that? Well, I think it could tell us a lot about how flexible cell identity might be, because you can imagine, how do you actually transform cells or tissue? This is sort of a big overarching question that I'm really interested in. So the animal I study this in is this strange little worm I told you about called Schizocardium californica. And I would like to hopefully posit to you that this is a perfect case study in transformation. So I've shown you the larva, I tell you it turns into the worm. How in the world does that happen? Don't worry, we'll walk through it. So essentially, the way we work with these animals is first we have to go out and find them. So like a lot of animals you might have in a lab, you can just order them online. We have to go find these in the wild. So this is in Morro Bay, which is about 2.5 hours south. So we dig these worms out of the mud. It's very stinky, so it's very sediment-rich mud. You have to go at exactly the right low tide. So at this point, the tide's coming back in, it's bad worm time. But if you're lucky, you'll get enough worms to do the research that you need to do. So in this case, we've just found a really lovely female that's full of lots of eggs, and that means we're gonna have lots of embryos to study in the lab. So once we get back to the lab, we cross our fingers, we hope that the female has eggs, and if we're lucky, a single female will release tens of thousands of eggs. This is actually quite common for these animals that are what we call broadcast spawners. They release a lot of embryos at one time. So we have these eggs in the lab, and now it's time for life to begin. It's a big responsibility to be parent to thousands of animals. I take it very seriously. So what happens after just a few short days is the larvae start to form. So this is an early larva. This is early in the larva's life. There's the top and the bottom of the larva, and there's its little stomach where it eats single-celled algae. As this animal continues to develop, they form this really intricate, beautiful shape and larval form that specifically has its own name. It's called the tornaria. And so this tornaria larva swims around in the water. It uses two main bands of cilia to do this that beat in the water. One band is used for feeding and the other is used for swimming, propelling the animal through the water. And then all of this is leading to that metamorphosis I was telling you about. So here during metamorphosis, essentially you have some bits that are a little more wormy and other bits that are, I don't know, a little still like the larva. So they still have that beating band of cilia, yet the top, the most anterior of the animals begin to condense into the head of the animal, and that will become the worm head. Let's see, I have one more. Yeah, so I told you about the swimming. It still has some of that larval structure, but a lot of this tissues began to transform into the adult worm. Yeah, and about 48 hours later, you have this thing, this little burrowing creature. This is now fully in its worm form. This is the hemichordate body. They are also colloquially known as acorn worms, and that's because the little head of the animal, the proboscis region, is thought to look a little bit like an acorn, so that's the acorn worms. They're their own phylum of animals. They're actually quite a unique group of animals, and they have sort of a body organization that's defined by 3 regions. They have a proboscis, a collar, and the trunk of the animal, and this spans from top to bottom. So given any particular time in lab, I may have animals at different stages. So we went collecting about 2-ish months ago, and so I have animals that are about 46 days old at this point. I showed you the video, so you don't have to look closely, but in this jar right here, I probably have, I don't know, a couple thousand larvae swimming around in this jar. So you can see, I don't know, it might just look like little spots, but essentially you can believe me that I have to take care of all these animals and carry seawater up the stairs into the lab. So we'll keep working with these animals and keep asking questions about them, how they develop, how they transform essentially into this crazy looking worm. I haven't told you all of the story, but essentially this animal, the way it transforms a distinct larva into the adult is through some combination of cell proliferation and cell death. And the thing we talked about today is sort of this change in cell state, like how cells actually transition their instructions from one life history to another. And really this approach requires a bunch of different things. It requires the organism biology to go out in the field and lead a research team to A, find animals, then B, you have to figure out how to take care of them, how to feed them. And then you use these technologies, say for encapsulating single cells into little tiny beads that then you can learn the information of. Then to the end, go back into the context of the animal and say, okay, cell, you had this information. Now, where are you? What do you do? How do you do that? That's sort of where we end up. I do think it's important for me to say that all of this work is hugely collaborative. I work with a really big research team. So I work with people both in my lab, but also throughout the Bay Area. So we're specifically supported by the Chan Zuckerberg BioHub. They support a lot of biomedical initiatives, but also have taken on weird animal questions like ours. So I'm happy to tell you more about our collaborations with a bunch of different groups throughout the Bay Area to accomplish this work, because it's a big project and I could not have done it on my own. And I am now happy to take any of your questions about these animals or other weird marine metamorphosis questions you might have.
Thank you, Paul. I see Stuart's got a question, but Stuart, I want to, before you ask yours, no doubt deep question, I've got a rather shallow question. I hope you'll allow it, Paul. It's about the term metamorphosis in general.
Sure.
Most of us, when we're hearing the word, perhaps first think of the caterpillar to butterfly example, which I think it's fair to say is a rather dramatic metamorphosis. In some of the changes that you described, it seemed like it was similar to calling human birth a metamorphosis. Can you help me be clearer about the definition of metamorphosis?
Yeah, so I think specifically the way we think about it is, we think about it through the lens a little bit of developmental biologists, because we're biased in our perspective. But specifically, we use the term metamorphosis where there is a distinct larval body plan in the way, essentially, for lack of a better term, but essentially a distinct body plan that has structures that are unique to the larva. So for example, those swimming bands of the larva that I work on are not there in the adult form. That makes it a distinct larva. We could talk about what structures are, say, distinct to a caterpillar that are not in the adult alone, right? So there are distinct structures that are sort of in between. But you're right in observing that that developmental process is in some ways very similar. And actually, I think the better correlation potentially is between, say, human embryonic development to how the butterfly embryo forms a caterpillar. Those processes, those early embryonic processes, very similar, but yet what they give rise to is quite different. Because what they give rise to in one case is the larva, in the other case is a full-formed adult. So maybe there's a interesting or not subtle difference.
All right. So with the exception of science fiction stories that involve shape-shifting humans, it is simply wrong to say that humans at any point in their development, from early fetus to adult, humans do not go through metamorphosis.
Not that I know of yet, though if it happens to someone, I would be all in for that. So.
Right. Then we've got a good sci-fi story. Right. Thanks, Paul. All right, Stuart, would you like to ask the question yourself?
Sure, thank you very much.
You bet.
Yeah, so the eggs that you collected, were they fertilized, or did you have to provide a male, or do it, have to be the surrogate fertilizer? And also, if you have time for the second question, were you able to somehow provoke the cells to convert from one type to another? Thank you.
Yeah, absolutely. So those are both great questions. So yes, these animals have separate sexes, male and female. I can usually tell them apart in the field, but often it takes coming back to the lab. So once we've separated them, the females get the best treatment. They get taken care of very well because they have to be under ideal conditions to release the eggs into the water. The eggs are released unfertilized. The males, on the other hand, we just rip open the sperm duct, take some sperm and fertilize the eggs, and we're good to go. So males are like dime a dozen. Females are very precious. It just applies generally. But specifically in this context, we fertilize the eggs. And that's true of a lot of marine invertebrates in general. So there are these broadcast spawners that release unfertilized things into the water. Though there are very fun, interesting, cool exceptions, which we could talk about more. But to your second question, have we been able to convert the cells fate? So the answer is no, and this system would be really bad to try it in because essentially we don't have a lot of the fancy genetic tools that you could say use in a lot of other classic research systems. I think the power of our study will be learning about what some of those switches could be that we wouldn't have known about otherwise, and then taking them into a system where you could manipulate. So for example, say being able to force a cell into a muscle cell fate, or a liver cell fate, or a, whatever cell fate you're interested in, we would learn what are the transition points to doing that. And then in a more manipulatable system, you could try something like that.
Good. Thanks for the question, Stuart. Let me get one in here again, Paul, if you don't mind. I can call your worm an acorn worm. That's not incorrect?
That is correct.
And so if I were to go to Morro Bay during low tide, would I have to have special skill to choose the right place to dig for an acorn worm?
Yeah, depending on when you asked me in my PhD, I would have had a different answer. My theories over the years have totally changed. Because I do think there is something characteristic about the burrows that these animals make, specifically the way they poop outside of the burrow. It's very distinctive. It looks like a little ice cream cone on top of mud. Now, the tricky part is other things can live in empty acorn worm burrows. So you can pick what you think is an acorn worm burrow, instead dig up a crab or a fish living in the mud. I don't know why they live in the mud, they live in the mud. So you can pick the right burrow. Whether or not there's a worm inside the burrow, that's up to you. And if you figure out something better, please let me know, because I spent way too much time digging up empty burrows.
Okay. Is it fair to assume that if I do go worming at Morro Bay, I should keep my eyes peeled for other low tide creatures to examine? You want to just name a few for us?
Yeah, there's all sorts of crazy things. There's some crazy, I think they're called ghost shrimp. They're wild. And then my favorite probably of the mud flat creatures is this giant nudibranch called Navinax. And what makes Navinax kind of wild is the only thing it eats is other nudibranchs. And the way it does this is by engulfing them in a single bite. So it's very, scary, but it's an amazing nudibranch. It's quite large. It's very colorful. Most of the animals in the mud flat are not necessarily very colorful. So this is a very colorful nudibranch that happens to be hanging out waiting to munch on other nudibranchs. So that's my favorite.
Yeah, but this is a small creature. It's not going to take on a dog or a human, yes?
I mean, as far as nudibranchs go, it's pretty big. It's like really big, which is a big nudibranch. Yeah.
Wow. All right, excellent. I see Jim's got a question. Please, if you'd like to ask a question yourself, make yourself visible, Jim, and let us hear it.
Okay, I hope I'm visible. So I was curious, you called these hemichordates. Mm-hmm. And if I remember now, this may have changed. I know things have changed quite a bit since way back when I was in school. I believe we humans are in the phylum of chordates. And I was wondering how closely related these little guys are to us.
Yeah, it's a great question. And you bring up an important topic, which is how animals are related to each other is constantly changing. So I could have told you something today, but yet there are new papers coming out every day, switching the positions of different groups, especially at these branching points that are a really long time ago. It's very hard to reassemble animals that diverged a long, long time ago. So you're right also to point out that the word hemichordate is suggestive of something. Well, it turns out at first scientists gave this name incorrectly. There was a hollow structure in the animal. They thought it was a nerve cord. It was not. It's just a hollow tube. So it's incorrectly called hemichordates. Yet we got very lucky because it turns out they actually do occupy a really important position in terms of the tree of life nearby the chordates. So they are invertebrate animals. They don't have a nerve cord. But they are this group of animals called Deuterostomes. You may have heard of protostomes and deuterostomes, and that just refers to what side perforates on the embryo first, your mouth or your butt. So essentially, this groups animals together. So we are grouped with hemichordates as deuterostome animals. So that means mouth second animals, so butt first animals. So we're actually related by that. So other deuterostomes include things like sea stars and sea urchins. Those are also invertebrate deuterostomes. Tunicates are invertebrate deuterostomes. And then we vertebrates also are invertebrate deuterostomes. So we often do think of these animals in the lens of evolution and asking what traits arose on the lineage to form, say, vertebrates.
Excellent, Paul, thank you. Do we have any more questions? All right, Jim, thanks for your question. I think we can draw this evening to a close. I want to thank everyone for being here. Please remember that our next Ask a Science Envoy event happens on March 30th, where we will learn about plant immune systems and imaging the planets around other stars, exoplanets.